Well, I don't know what you tell people when you get on an airplane and they say, what do you do for a living? So I like to tell people I'm a professor and I do research, teaching, and service. And if I really want to talk about my research, what I don't do is tell them I work on polyploidy and the origins of novelty, impact of duplications of genome and network evolution. Instead, what I tell people is I work on the dogs of the plant world. And the reason why I say I work on the dogs of the plant world is because everybody loves their dog, and everybody knows that a Chihuahua and a Great Dane are one species. But not everybody knows that Brassica oleracea, cabbage, broccoli, cauliflower, kale, kohlrabi, are one species. And I can tell them all about the mini cultivars and how they're really diverse and how they have most of their parts have been domesticated above ground and below ground. And then we can spend the next half hour talking about their dogs, which is great. So then you can just be like Bill Nye the Science Guy. If we go a little deeper, I'll tell them another fun fact, like did you know that in Asia there's another dog of the plant world, Brassica rapa? So turnip, rapini, Chinese cabbage, bok choy, field mustard, yellow sarsenet, and brown sarsenets are also one species. And in a way, they're more impressive than Brassica oleracea because they've been domesticated not only as above and below ground vegetables, but also as oilseeds. And if I'm feeling really adventurous, I'll tell them, did you know that the tradition of carving pumpkins in the US came from Europe, where they used to carve turnips and Swedes and rutabagas and sugar beets. And when they came to the US, they said, forget that, pumpkins. Those are huge, they're really fun to carve. Like, who wants to make one of these? So today I'm not gonna talk about the evolution of those groups. We just had a couple papers published with Mike Barker and Mike Gore and others on how those plants are all related to one another. But I want to focus on polyploidy. So first I want to define polyploidy. So polyploidy is when a nucleus containing 3 or more copies come together in a genome. So you know our sperm and our eggs are haploid and we are diploid. And then you can double that and be tetraploid. There's some other words you need to know. So autopolyploid is a term we use when you have 2 genomes that merge together from the same species, so like genomes A and A prime, come A and A prime together. But what I'm going to talk about today are allopolyploids. And these are when you have 2 separate species, for example, Brassica oleracea and Brassica rapa, that merge to become Brassica napus or canola. So one of the big surprises of the genomics era is that we now know that many things that are diploid are actually ancient polyploids. So when you look down the chromosome at microscopes, or look down a microscope at chromosomes of corn and other things, and you see multivalent pairing or things going on, people often suspected, based on this and markers, that corn was a paleopolyploid, and we know that that is true relative to sorghum due to genomics. And in the best figure of the Arabidopsis genome paper, the biggest surprise of the whole Arabidopsis genome, because it's got a small genome and only has 5 little chromosomes, is that it has not 1, not 2, but 3 whole genome duplications in it, and actually more if you dig in deeper. And so, we used to say things like, well, 70% of flowering plants are polyploid, because if you look at the tips of the tree, it's true. Most of the plants are currently diploid. But if you go back far enough in evolutionary time, you can see that actually all flowering plants are polyploid. And so when Jim Lehmannsbach is here and colleagues sequenced the Ambrella genome, they found that it showed signatures of polyploidy and that all flowering plants are polyploid. And similarly, you and I are polyploid because all vertebrates are polyploid. And fish are polyploid. And JGI just sequenced Xenopus, which is polyploid. We know a lot of these insect groups are polyploid. So really, it's not a question for a lot of eukaryotic lineages of, are you polyploid? It's, when were you last polyploid? Oh, I was last polyploid several 100 million years ago, right? And what are those duplicate genes doing? So a lot of plant groups have gone crazy with polyploidy and use these duplicate genes to do really cool stuff. So if I start on the left side of the phylogeny, and I just told you that Arabidopsis had 3 or more ancient polyploid events relative to Ambrella, or the ancestral flowering plant genome, it is actually 12x. And then if you have a whole genome or a hexaploidy event, kind of like wheat, but it was 20 million years ago, the crop brassicas, the diploids now, Brassica and oleracea, are actually 36x. And then when you merge rapa and oleracea to get Brassica napus, you have a 72x genome relative to the ancestral flowering plant genome. And so there's a few other terms you need to know. So these recent polyploid events where you can still see the parental chromosomes, we call neopolyploidy. And these polyploid events where there's still large segments of chromosomes that show polyploidy and affect pairing, we call mesopolyploidy. And these really, really old events where we just have duplicate genes left over, we call paleopolyploidy. So why do we care about polyploidy for those of us who work in crop plants? Well, it turns out, this was a paper that was published last year, that most domesticated plants have been preceded by a polyploid event. And if you look at the genes that are domesticated, those are duplicate genes that came out by polyploidy. So why and how does that work? So way back in 2004, when I was a postdoc in Wisconsin, I did this experiment with Eric Schranz, where we took a Brassica rapa and a Brassica oleracea inbred lines, and we crossed them, and we made one polyploid Brassica napus. And then we selected them over time for early and late flowering. And within 6 generations, we basically had 2 species. One, if you threw the seeds out in red, flowers in 30 days or so, and the other one in blue that would flower later. So if you put these plants outside, they wouldn't even see each other sexually. So how is it after so few generations you get these big phenotypic effects? So in the polyploid literature, there was lots of things at the time about dosage or maybe epigenetics, but people really weren't sure what could cause this. So we discovered a phenomenon which I think is pretty widespread, and I want to share it with you. So this is my most important slide, so I put it on a blue background. So on green are chromosomes of Brassica rapa, and they have the same kind of chromosome exchange that you and I have, homologous chromosome exchange. Now when rapa in green and oleracea in yellow merge together in Brassica napus or canola, you can now have exchanges between the 2 parental genomes. And to indicate that, we add more vowels. So we now call it not homologous exchange, but homeologous exchange. And you can draw the Punnett square, but as a result of these reciprocal exchanges, you can end up with situations like this, where you have a green chromosome with a little bit of yellow on top, yellow with a little bit of green on top, or more exciting is the non-reciprocal versions on the right. The exchanges are reciprocal, but the modes of inheritance lead them to be non-reciprocal. So now let's imagine the phenotype that I just told you about. Imagine the big blue circles are early flowering time, and the little circles are late flowering time, and Brassica napus has a mid-parent value for that flowering time, so 30 days, 60 days, 45 days. Now you have one mutation, one chromosome rearrangement, and you can have all early flowering time or all late flowering time. And it turns out if you look at the genes that are on these chromosomes, there are things like flowering locus C, constans, there's like a whole group of flowering time genes. And at the time in the literature, people like Hopi Hoekstra and others were talking about speciation genes, right? The gene for speciation. And so we were calling these, speciation rearrangements. Well, a lot of people didn't really believe this result because they said this could be due to epigenetic or gene loss or whatever. So then we really wanted to show it on steroids. So Lauren Riesberg was becoming famous at the time for replaying the evolutionary tape 3 times with resynthesized Helianthus hybrids. So we decided to make 100 lines of Brassica napus, which we did. But I can only put 50 on an RFOP block back in 2005, 2006. And so we did this experiment where we took a small Brassica rapa plant that was rapid cycling, a small Brassica oleracea that was plant that was rapid cycling, made 50 of these lines. They're hugely heterotic. The canola plants are twice the size. And when we first phenotyped them and genotyped them, they were all the same. 1 + 1 = 2. But after selfing them for 5 generations, they were rapidly diverged. And we've now selfed them to the S10 generation, and we would really like to sequence these so know more about them. So what happened with these lines? Well, you have huge variation, much larger organ size, crazy morphological variation, flowering time variation, and now we're going deep into metabolism. And I just show you a family portrait of one of these slides. So on the left is the S1 generation, and you can see by the 5th generation, there was a rearrangement that caused it to become a dwarf line. So at this point, people said, okay, we now believe you, these homologous exchanges occur, but they don't occur in natural Brassica napus. These are just your weird mutant plants. And so I was happy 10 years later when we published the Brassica napus genome that we found them in natural Brassica napus. So this is just one example. So this is chromosome R1 and O1 are from the ANC genome. And on the Brassica napus genome, I've taken the homologous exchanges and painted in this cartoon what we found. So we found these big blocks of both reciprocal and non-reciprocal exchanges. And actually, we found 17 of these segments all over the Brassica napus genome. And do these matter for phenotype? Well, they do. So we also sequenced a diversity set of these across spring oil seeds, fall oil seeds, rutabagas and kales. And you can see here, here's a rearrangement on chromosome C2 and A2, which affects glucosinolate. So you know canola is called canola Canadian oil because it has changes in glucosinolates. And these rearrangements also affect traits like disease resistance, oil biosynthesis, and a lot of things that JGI, for example, would care about. So these are, just to make the point, these are changes that don't necessarily change DNA sequence, A, C's, T's, and G's, but are moving large blocks of genetic material around. So it's presence-absent variation or copy number variation, the same kind of variation that we're now seeing all over the human genome. So my take-home point number one is that by studying, you can find things in these resynthesized lines by experimental evolution or look at natural variation that could be found in crop plants. So after we found them in Brassica, people said, well, that is only in Brassica. And then it was found in wheat, and then in Tragopogon. And recently, Scott Jackson has reported something like this in peanut. And so because we were able to see some of these events by looking at resynthesized lines and other kind of examples of experimental evolution, I think we're kind of prepared mentally, so when we see these things rarely in crop plants, we can recognize them for what they are. And I've been talking with Tom Younger and others about what they're saying in panicum, switchgrass. There's some unpublished reports in rice, some things going on in legumes, so I know that these are actually more common than people think. Or they mistakenly call them gene losses or some kind of conversion that they don't understand. So for the second part of the talk, I want to talk about mustard. So another airplane thing I ask people is, well, why do you think plants have the taste that we like? Is it for us or whatever? So that strong, sharp taste of mustard, wasabi, horseradish, where does it come from? Well, it comes from glucosinolates. And so these are compounds that the plant uses to defend itself against insects and other herbivores. But some insects, like the cabbage butterfly, now prefer to eat these plants because they have figured out how to defuse the mustard oil bomb. But this isn't necessarily good for the caterpillar. Well, it's good for the caterpillar because they can nom, 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 eat the plant. But then there's some wasps that also key in on these glucosinolates because then they can lay their eggs on the caterpillars and mummify them. So this is a paper that Pat Edger, who's a grad student in my lab, who's now a professor at Michigan State University, wrote and defended his PhD in 2012, and we're about ready to publish the plant side of the story when Chris Wheat, who studies the cabbage butterflies, was driving through Missouri on his way to the Genomics meeting in Kansas City, and we lined up our plant phylogeny with his insect phylogeny and said, okay, this is too good to be true, so let's combine these two stories. So that took 3 years to do that. And so this got a lot of press, so NPR, Washington Post, Radio New Zealand, Daily Mail, but I knew we hit on something big when we made the opening joke of the Big Bang Theory. So I think I can play this.
Could you pass the mustard?
Sure. Hey, want to hear a fun fact about mustard?
Is it that the glucosinolates, which give mustard its flavor, were evolved by the cabbage family as a chemical defense against caterpillars?
Yeah.
Well, that was fun. Good for you, Leonard.
So I didn't care about any awards or anything else after this was on TV. I figured, okay, I can drop the mic, right? Yeah. Awesome. Okay. The other thing, so this paper also was conveniently 50 years after Ehrlich and Raven published Coevolution, which they described in 1964. And their main example for coevolution was indeed cabbage butterflies, or the interaction between the caterpillars and the plants that they eat. And they talked about this in terms of an arms race. And so the idea is this, that plants evolve chemical compounds that allow them to fend off herbivores, and the plants then are successful, more successful than their sister groups that don't have those compounds, similar to what Gloria was talking about yesterday with adaptation of some plants onto dry places. And then the insects figure out countermeasures and figure out how to get around some of these compounds. So now I got a happy caterpillar who's ready to eat this plant. And this goes back and forth. And eventually, the caterpillars actually hone in on the plant defense as a way to find them. And so what Pat Edger in my lab did was this. So we took a lot of transcriptomes that we made ourselves in our lab and used these and anchored in genomes to make a very large phylogeny for the Brassicales, which had not been resolved before. And so the tree is on the left side. And then we were able to figure out and order how the compounds of the mustards had evolved. And that hadn't been done before either. And then finally, we were able to place where these whole genome duplication events had been. Because in the literature, people had long thought that these events that were seen in Arabidopsis were really, really old. And we found out that they were actually much, much more recent than had been known before. And so you can see where this blue diamond is, where this beta and alpha event on the tree, are where we found these polyploid events to be. And then the red stars on the tree are where the species really started to radiate. So if you've taken a plant systematics class, you've probably never heard of some of these families like Coevolution and things like that because they only have 1 or 2 species. But when you get all these glucosinolates piled up, you now have big families that you have heard of, capers, cleome, the mustard family. And we found that after these polyploid events, there was a lag time to when these new metabolic traits. So then you have a mustard bomb, and now you have a fancy mustard bomb, and now you have a really fancy mustard bomb, and now you have a super fancy mustard bomb, and Brassica have 4,000 species. And so what we did with Chris Wheat was we took his butterfly phylogeny, which is on the bottom here, and we lined it up with fossil data that we had from both the plants and the insects. And so what you see in blue is where the events line up for the origins of the plants getting attacked by these butterflies. So the butterflies used to feed on legumes and relatives, and now they moved on to the Brassicales. The plants evolved indolic glucosinolates, and then the butterflies start recognizing these glucosinolates as something to hone in on. And the mechanism that they get to detoxify the mustard oil bomb is an NSP. And then at this time of the tree, in green, the butterflies shift onto these new plant families, Caperaceae and Cleomaceae. There's a massive species radiation on both the butterfly and the plant side, and there's novel chemicals on both sides of the chemical warfare. And in the third round of this arms race, there's a shift in the Brassicaceae to even more complex compounds, and there's 2 independent lineages of butterflies that move on to these guys. So the second big point of my talk is this. So I wake up in the morning thinking of diagrams like this. You take your favorite omics or your favorite network or your favorite web and think about what happens after a polyploid event. You duplicate everything. You duplicate everything. And then you can lose parts, but you've duplicated everything. So you have a system that can work. So I like to say when I do systems biology, we have the best system question, which is what happens when you double the whole system? What are the new networks that can evolve that can lead to novel phenotypes, crop improvement, or opportunities for synthetic biology? And my thought partners in this have been Gavin Conant, who's now at NC State, and Jim Birchler, and here's a citation for a paper that we've written about this. Now with JGI, we've had a very long-term project on looking at broader across the Brassicaceae. So we call this the Brassicales Map Alignment Project, or the BMAP project. And there's about 15 genomes that have been out there for the Brassicaceae that have been published, and they're in blue on this tree, and we're adding 19. So you can see we really, really have expanded what we're going to know about the Brassicaceae. So these genomes have been assembled, we have these trees put together, and I think there's going to be a lot of good stories that come out of this because, for example, we have convergent evolution of oil seeds, extremophiles that occur in all kinds of crazy environments, polyploidy that occurs all over the place so we can replay the evolutionary tape deeper in time. And for me, this has been great fun because I feel like we put together this dream team. Rod Wing and Detlef Weigel got this started when Rod was out of sabbatical in Germany. And then Stephen Wright, Tom Mitchell-Olds, Eric Sharons, and I, and others put this together. And the people really doing the work are folks like Tyler Kent and Pat Edger and Jacob Washburn and many others. So my second final point here is that I think we can use these comparative genomics and network approaches to really think about possible rules for synthetic biology. So we used to use terms like neo-functionalization and subfunctionalization to talk about genes, and now we can use those to talk about pathways. If I'm talking to a bioinformatics crowd, the first thing I tell them is, you just sort out duplicates. It's actually really important to sort out polyploid duplicates versus tandem duplicates because they evolve different ways. They evolve by different rules, if you will. And then finally, I've been thinking a lot recently about metabolism. And I've been really inspired by the paper that Jen Weisskaver, sitting over there, put on bioRxiv recently about how specialized metabolism may evolve, because it's different than you may think otherwise. So I've been generously funded over the years by NSF. I've been working on a project right now with Mike Barker and Eric Lyons and Gavin Conant on the Brassicas. I'm working with USDA and Cornell in New York on Brassica rapa. I just got a DOD grant to use Brassica as a low-dose radiation sensor. You figured that out. And then with JGI, we have the BMAP project, which is really ongoing with all the people that I just talked about. And hopefully next year, the year after, my student Sarah Unruh, sitting over there, who's crushing Twitter at Sow916, and Francis Martin, who I think is here somewhere, he's also crushing Twitter. So we have a collaboration to look at orchids and orchid fungi, and that's going to be something we'll talk about later. Okay, thank you.
Thank you very much, Chris. People often ask at review panels or advisory boards, what keeps you up at night? And I love the way Chris said it. What I wake up in the morning thinking about is this. It's a much more positive spin on things. Are there any questions?
Oh, good. It was all perfectly clear.
Hi. In your example, you showed a lot of tetraploidy. But what effect during those 6 generations, when you get to like a dwarf plant or different other types of plants, do trisomies and other polyploidy have during those 6 generations?
So I didn't show it because it would be an hour talk, but we see not only chromosome rearrangements, but chromosome replacement. And it turns out the plants that are happy can gain and lose a chromosome. So you may look under the microscope and you still see 24 chromosomes, which is supposed to have, but there'll be chromosomes that have been lost and gained. And as long as they're equal number, they'll keep going. But if you get trisomics, they don't make pollen and they're really unhappy. So we talk about this as a polyploid ratchet. You want to have some rearrangements, but not too many. Otherwise, meiosis goes out of control.
How close to chromosomes? Sorry, how close do chromosomes have to be in the brassicas to be able to support a homologous recombination?
Good. So it's true that rapa has 9 chromosomes. Rapa and Oleracea are different that they have 9 and 10 chromosomes. So obviously not all of them pair. But the 3 largest chromosomes are completely synthetic, and so those are the ones that most frequently form multivalents, and those are the ones that undergo most of these rearrangements. So the other pairs of chromosomes much less.
So bigger chromosomes, more crossovers, more chance of this
absolutely.
Kind of thing happening.
And we found that also in both in the resynthesized and the natural lines of Brassica, and I think wheat shows the same pattern.
I had a question about one of your first slides and your last slide. So you were posing the question of not are you polyploidy, but when were you polyploidy? So if you take that second question, then there's the assumption that there must have been genome condensing at some point.
Absolutely.
Now, if you think about the idea of genome duplication leading to neofunctionalism and subfunctionalization, can you think of the opposite of being true? That once you come to this condensed state, I don't know how the mechanism would work, but maybe through alternative splicing somehow on recombination, that those 2 duplicated functions or those 2 duplicated genes now in the non-polyploidy state are now an alternative splicing form or something like that.
So what's the question, actually?
So I guess the question is, when you have loss of polyploidy,
Yes.
is there an advantage to that from the point of view of, we always think of, if you have more gene duplications, you must have more functions, or the possibility of functions, and by losing those proteins, you're losing function. Perhaps there's a way to save that.
Yeah. The theory that we've put out there is that if you have a tandem duplication, you can't create these elaborate complexes by tandem duplication, usually all the horizontal gene transfer and things. But if you have a polyploid event, if you have things in cis and trans, it doesn't matter because everything is stable. And so as you lose genes, as long as they're not deleterious, you can dipletize. But you've got to actually keep things that work in these stoichiometric configurations, like transcription factors, whatever. And so there's a ton of literature on the bias of which genes are kept just based on Go categories. So what I'm interested in is as you have more ploidy events, does that get erased? When does the meiosis turn over in these plants? And certainly there's a lot of people who are interested in alternative splicing as a way to preserve duplicate genes, but there hasn't been a lot done on how people get rid of genes, to my knowledge, as far as mechanism. Yeah, Gloria.
We have one here too.
Oh, sorry, wherever. Matt.
Sorry. Hi, Chris. I had an argument with a phylogenetics, bioinformatics person recently where she didn't understand why we have to separate homologous genes from just regular paralogs.
Yeah.
And obviously they arose in different ways, but taxonomically, you know, she argued that they were exactly the same thing.
No.
And I find it hard to put my finger on exactly why. I thought you might like to answer that one.
Sure. Well, I'll kind of state what I did before. You've drawn, so Mike Freeling, and this isn't just in plants, actually. This is in yeast and paramecium, whatever. A lot of people have looked at the fate, what's that? And frogs, because, you know, Xenopus. And salmon. Like, so people looked across at the fate of duplicates that are these paralogs versus the homologs, and they're very different, what they end up doing and what kind of genes are retained. I'm actually kind of interested in how they move around also, but they certainly have different, like if you sort out things by function and what they end up doing, it is absolutely different between the homologs and the paralogs. I can send her lots of papers or whatever. But yeah, they're definitely not the same. The natural history of them is different.
So bringing a question back to the dog versus plants. First of all, I wouldn't want to sit next to you on a plane because I never want to talk to anybody on a plane. Okay, now the serious question. So both dog... No, I'll sit next to you. We can talk about
Thank you. Okay. I was a little worried. I'm like, we could talk about transcriptomics, right?
Talk.
Come on, let's bring it on. Okay.
You're definitely cool. So dogs and plants have both been domesticated, bred, right?
Yeah.
And so I'm just curious about the polyploid issue. Is it something linked to domestication in plants and breeding in dogs? I'm really not clear how it weighs out in other organisms like even us.
Exactly. So I took it out of my talk because we haven't published it yet. But if you just look across these dogs, these plants, brassicas, and you see which genes are under selection, and then you ask what are those genes that are under selection, and what's their history? Back to Matt's question. Oh, they often are duplicates. What kind of duplicates? Oh, are they tandem? No, they're polyploid duplicates. And so if you expect these genes that are under selection under a certain frequency to be selected for, they're way more than by chance the genes that were created by polyploidy. And so at least in these plants, like the targets of selection are these duplicates. And so what I've noticed is people like me who started with comparative genomics and comparing species are now running to do population genetics. And people like Ed Buckler and I think Tom Bretnell and others are moving from doing more PopGen and Maze or whatever to, oh, now I'm going to compare things across C4 in different species or whatever. And so I think Ed Buckler wants to sequence all the andropogony. So now we're talking to each other. Well, what insights can we get from both kind of studies you do across species to ones within species to see what these kind of rules for synthetic biology would be. So.
Thanks. If there are no more questions, thank you very much, Chris.